Hello, and thank you all for being here today. My name is Katelyn West, and I'm a rising fourth-year medical student at Western University of Health Sciences in Lebanon, Oregon. This research was made possible through the support of the Medical Anatomy Center. My presentation is titled Advancing Brain Vascular Big Data Using BriteVu Perfuse GAXspecimens, a novel approach to high-resolution neurovascular imaging and anatomical analysis. Today, I'll discuss an emerging technique that may improve how we visualize and study the cerebral vascular system. This work explores how combining BriteVu vascular perfusion with GAX cadaver specimens can generate highly detailed neurovascular imaging data sets beyond the limits of conventional clinical imaging. The goal of this presentation is to explain why current imaging techniques have limitations, how this approach may help overcome some of those challenges, and why it may have implications for neuroscience research, surgical education, neurointerventional planning, and future AI-driven technologies. Big data in medicine refers to the enormous data sets generated from imaging studies, electronic records, and genomic sequencing, as well as physiologic monitoring and healthcare systems. These data sets often require advanced computational tools, such as artificial intelligence and machine learning for analysis. The goal of big data in healthcare is to improve patient care and clinical decision-making by identifying patterns related to disease risk, treatment response, and outcomes. In neuroscience, imaging data is especially important. Neuroimaging datasets are increasingly used to train algorithms for stroke detection, tumor classification, vascular segmentation, surgical planning, and automated image interpretation. However, AI systems are fundamentally dependent on the quality of data used during training. If imaging lacks sufficient vascular detail, computational models may also lack anatomical precision. As medicine moves towards precision medicine and AI-assisted diagnostics, there is growing demand for higher resolution anatomical datasets. The brain's vascular system is extraordinarily intricate and essential for normal neurologic function. Because the brain has very limited tolerance for ischemia, detailed understanding of cerebral vascular anatomy is critical across nearly every neuroscience specialty. In neurosurgery, injury to even the small perforating vessels can result in devastating neurologic deficits, making vascular anatomy central to procedures such as aneurysm surgery, skull-base approaches, AVM treatment, and tumor resections. In neuro-oncology, tumors frequently distort or recruit abnormal vascular supply through angiogenesis, making vascular understanding important for preserving normal perfusion and minimizing complications. In stroke neurology and interventional neuroradiology, detailed vascular knowledge is foundational for thrombectomy, aneurysm treatment, and catheter-based procedures performed within millimeters of critical neural structures. Importantly, many clinically relevant vessels are extremely small and fall below the resolution limits of conventional CT or MR angiography, leaving much of the brain's microvascular anatomy incompletely visualized. Improving neurovascular resolution, therefore, has several important clinical implications. Although CT and MRI technologies have dramatically advanced neuroscience, importantly, limitations still exist when imaging the cerebral vasculature. One major challenge is motion artifact. Even subtle patient movement or physiologic pulsation can degrade image quality, particularly when evaluating very small vessels. Another limitation is contrast timing. CT and MR angiography require image acquisition during a narrow window after contrast injection, and patient-specific circulation differences can affect vascular opacification. Safety constraints also limit optimization in living patients. Radiation exposure, nephrotoxicity, scan duration, and contrast-related risks restrict how aggressively imaging protocols can be performed. Resolution remains another major issue. conventional imaging generally visualizes vessels around 0.7 to 1.5 millimeters in diameter, while many clinically important perforators and microvascular structures are smaller than this threshold. As a result, conventional imaging often prioritizes diagnostic practicality over complete anatomical mapping. This study explores whether postmortem vascular perfusion models may help overcome some of these limitations. The objective of this study was to determine whether BriteVu perfused GAXspecimens could generate higher resolution neurovascular imaging datasets than conventional imaging methods. We investigated whether combining BriteVu contrast perfusion with advanced postmortem specimen preparation and multimodal imaging could improve vascular density detection. The study incorporated CT, MRI, and micro-CT imaging, each providing different advantages in spatial resolution and vascular visualization. The hypothesis was that because these specimens are not limited by physiologic circulation, patient movement, or contrast timing restrictions, the vascular system could be perfused more completely and imaged in greater detail than is possible in living patients. BriteVu is a specialized radiopaque contrast agent designed for vascular perfusion imaging. Unlike conventional intravenous contrast used in live patients, BriteVu is optimized for postmortem vascular filling and can penetrate much smaller vascular structures. One major advantage is its ability to perfuse small arteries, venules, and capillary level structures that may not be adequately visualized with standard imaging contrast agents. Because the specimens are postmortem, perfusion can occur under controlled conditions without concerns related to nephrotoxicity, contrast reactions, or circulation timing limitations. The result is a highly detailed vascular cast that can then be imaged using CT, MRI, or micro-CT techniques. GAXspecimens are a novel anatomical preparation model designed to preserve lifelike tissue characteristics while maintaining vascular integrity. It does not use formaldehyde, phenol, or salt solutions. It is a water-based system which is minimally toxic to the handler and the environment. GAXspecimens are stored in refrigeration for a minimum of 6 to 12 months, and they can be used for dissection as well as procedural and surgical training. Real or artificial blood can even be perfused through a GAX vasculature. With intermittent positive pressure ventilation and pre-mortem functioning lungs, oxygen exchange can occur and can be measured with pulse oximetry. They can be intubated and with intermittent positive pressure ventilation can actually breathe. GAX specimen kidneys continue to produce a urine-like fluid if renal function was intact pre-mortem. Unlike traditional cadaver specimens, these preparations preserve tissue flexibility and anatomical realism while allowing vascular perfusion imaging. Another major advantage is multimodal imaging compatibility. These specimens can undergo CT, MRI, micro-CT imaging, and also anatomical dissection afterward, allowing for direct correlation between imaging findings and physical anatomy. Overall, GAXspecimens provide a bridge between traditional cadaver anatomy and modern, high-resolution imaging research. This study combined literature review, specimen preparation, multimodal imaging, and anatomical dissection. A literature review was first performed to establish the typical vessel diameter ranges visualized using conventional CT angiography and MR angiography. The experimental portion involved vascular perfusion of GAXspecimens using BriteVu contrast material. The first experimental group consisted of 25 specimens, representing 50 anatomical sides that underwent BriteVu perfusion, followed by CT and micro-CT imaging. A second group consisted of 10 specimens, representing 20 anatomical sides that underwent perfusion with both BriteVu and gadolinium to optimize MRI visualization. Following imaging acquisition, detailed anatomical dissections were performed to correlate imaging findings with physical anatomy. The results demonstrated substantial improvements in vascular visualization compared with conventional imaging techniques. Traditional clinical imaging generally visualizes vessels approximately 0.7 to 1.5 millimeters in diameter, while structures below this threshold are often poorly visualized. In contrast, the BriteVu perfused GAXspecimens demonstrated markedly increased vascular density and improved visualization of smaller vascular structures. Importantly, all specimens were successfully prepared and imaged, resulting in a 100% preparation success rate. The data sets demonstrated enhanced visualization of smaller arterial branches, venous structures, and dense vascular networks that are often difficult to appreciate using standard clinical imaging alone. These findings suggest that this preparation technique may significantly expand the amount of anatomical information obtainable from neurovascular imaging datasets. There are several major differences between conventional live patient imaging and GAX plus BriteVu imaging approach. As I have previously mentioned, our perfusion model eliminates motion artifact and allows controlled vascular filling without physiologic timing limitations. The perfused specimens demonstrated much greater vascular density and visualization of the smaller vessels than conventional imaging typically allows. These differences help explain why post-mortem vascular perfusion models may provide uniquely valuable datasets for anatomical research and AI development. One of the most exciting implications of this work involves artificial intelligence and machine learning. Modern AI systems depend heavily on the quality of the data used during training. In neuroimaging, machine learning algorithms are increasingly used for stroke detection, vessel segmentation, tumor analysis, and surgical planning. However, if imaging datasets are incompletely representing the cerebral vascular anatomy, the resulting models may also have limited accuracy. Higher resolution vascular datasets could improve AI performance by allowing algorithms to learn more detailed vessel branching patterns, as well as collateral circulation and microvascular organization. These datasets may also enhance surgical simulation technologies. As medicine continues moving toward computational neuroscience and precision medicine, the demand for richer anatomical datasets will likely continue to grow. The applications of this technique extend beyond anatomy research alone. In neurosurgery and neurointerventional training, these models may improve understanding of complex vascular anatomy and support more realistic surgical simulation. For stroke education, detailed datasets may improve visualization of collateral circulation, vascular territories, and perforator anatomy. In medical education, high-resolution vascular imaging and 3D reconstructions could enhance spatial understanding of cerebral vascular anatomy beyond the traditional atlases. From a research perspective, these models may also support studies involving vascular pathology, tumor angiogenesis, and microvascular disease. Despite the promising findings, several limitations must be acknowledged. First, this is a deceased donor model rather than a living physiologic system. Although the specimens provide excellent anatomical detail, they cannot fully replicate dynamic blood flow or vascular physiology present in living patients. Second, the study sample size remains relatively limited, and larger studies will be needed to validate reproducibility across broader anatomical populations. Standardization of perfusion pressure, contrast distribution, imaging protocols, and specimen preparation techniques also remains important. Finally, while these datasets may improve anatomical understanding and AI development, it remains uncertain how directly these findings can be translated into live clinical imaging. This work opens several promising directions for future research. One major goal is development of larger standardized neurovascular imaging datasets to support AI development and computational anatomical modeling. Further studies may also incorporate 3D vascular reconstruction, virtual reality simulation, and comparative pathology models involving stroke, tumors, aneurysms, and microvascular disease. Advanced datasets may improve automated vessel segmentation, anatomical labeling, and predictive modeling for cerebral vascular disease. Ultimately, this work may contribute to a more detailed digital understanding of the human cerebral vascular anatomy than has previously been possible. Vascular imaging could even potentially serve as a unique biomarker, allowing patients to be grouped based on shared vascular characteristics and stratified by risk to guide earlier intervention. For example, variations in the circle of Willis and other vascular patterns combined with machine learning may help identify subtle imaging signatures associated with future stroke risk, enabling earlier recognition of high-risk patients. In conclusion, this study explored a novel approach for improving neurovascular imaging datasets using BriteVu perfused GAXspecimens. Conventional vascular imaging remains limited by motion artifact, contrast timing, patient safety considerations, and restricted spatial resolution. By combining advanced vascular perfusion with multimodal imaging in deceased donor specimens, this approach demonstrated improved visualization of smaller and denser vascular structures beyond many conventional imaging thresholds. These findings suggest that advanced perfusion imaging models may significantly enhance neurovascular anatomical datasets with potential applications in AI development, surgical simulation, neuroeducation, vascular mapping, and neuroscience research. As medicine increasingly incorporates artificial intelligence and precision imaging technologies, the quality of anatomical datasets becomes critically important. Thank you all very much for your attention and time today. I'd be happy to answer any questions.